topic this week is going to be the study of whole genomes, or genome sequences. And as is often the case, I want to start by talking a bit about some of the technology that goes into finding these. So how do you get whole genomes? And then in the next class, get into some of the work that goes into how we computationally analyze these, what's called annotation of a genome. And then on Friday, get into the hands-on issues of how you access genomes, what kinds of information you can get out of them, so you can use them in your own analyses. So this is going to be a bit unusual in the more biotechnology-focused lectures we've talked about, in that the biotechnology behind genome assembly is actually very computational itself, so you really can't get a whole genome without a significant amount of computation. So I want to talk just a fair amount today about the computational issues that go into getting entire genome sequences. Before we get to that, though, I want to say a little bit about why we care, so why we want whole genomes. There are a lot of ways genomic sequences are being used in modern biological analysis, but a few of them I just mention up here. One reason we're interested in whole genome sequences is a reason that I mentioned for being interested in a number of different biological data types, and that is studies of evolution. So genome sequences are often used in their entirety, or at least in large portions, to study evolution, because on very long time scales, we can map evolution, or at least measure evolutionary distances approximately in terms of rearrangements of large regions of genomes. So rearrangements of entire genes, or duplications or losses of genes. These tend to be the best evidence we have for very long time scales of evolution. So if you're trying to build evolutionary trees on scales of hundreds of millions of years, say. So this is one of the reasons this kind of information is useful. The more whole genomes we have, the more accurately and completely we can do this. Another reason we care quite a bit about whole genomes is because a lot of important information about an organism is found outside of the protein coding regions. I think there was maybe a naive view before whole genomes became available that everything interesting is found in the genes, that more or less an organism is a collection of genes that code for proteins. And I don't think anyone who worked on genomics ever believed that that was completely true, but you might have heard the term that most of our genomes are junk DNA. And I think the more we look at these, the more we appreciate just how much is going on outside of the genes. In some cases, this was the discovery that non-coding genes, so genes that code for RNA sequences that are directly functional, are much more common and much more important in large eukaryotes like us than was appreciated before we had a genome sequence. And then there's just a lot more information that's encoded in a genome regarding how the genome is regulated, how it's structured, how that structure changes under different kinds of conditions. So basically, the more we look, the more we learn about genome sequences. Some other uses of these are very important for specific medical contexts, and some of this we'll hear a bit about later in the term. So identifying genotype-phenotype correlations is one of the important reasons people study genomes. You want to be able to figure out that there are variations in particular genes or regions of a genome that are, let's say, correlated with risk of a disease that might help us find genes that might be good targets of drugs to treat or prevent those diseases. This is something that depends very critically on having a good map of the entire genome, knowing approximately where things are relative to one another in the genome. So that's one of the kinds of studies that have really been strongly facilitated by having genomes available. This also has some special uses in studies of cancers, because genomic scale changes are very commonly a way that cancers develop and progress. So cancers rearrange large pieces of their genome, they gain or lose copies of pieces of the genome, and these things are very hard to study if you don't have a good model of what the genome looks like. So that's another special medical context where it becomes quite valuable to have entire genomes and to be able to compare entire genomes. So this is far from an exhaustive list, but is some of the motivation behind why we want to do this. Any questions about this? All right, so that brings us to how we get genomes. You may remember back in the first week of the class, we talked a bit about sequencing technologies. If you want to sequence a genome, you need to be able to sequence DNA. But the sequencing technologies we saw are only able to sequence small pieces of DNA. So the automated sequencing machines we looked at might be able to give us lots of sequences of individual pieces of DNA, but each of those individual pieces is short. Depending on the technology, it might be up to about 1,000 bases, 1 kilobase of DNA. And that creates a problem if you care about whole genomes, because whole genomes are much bigger than the things our genome sequencing machines can sequence. And a lot of the question I want to deal with today is how we go from what we had seen back in week 1 to get, let's say, a kilobase of sequence to an entire genome. Even a very simple genome like a virus, you might be talking at the low end on the order of 10 kilobases of DNA. A prokaryote, like an E. coli, for example, we would be talking on the order of megabases, so millions of bases of DNA. And a complex eukaryote like us, you would typically be talking in gigabases of DNA. Some of the more complicated ones, like plant genomes, tend to be big. We might be talking hundreds of gigabases of DNA in a complete genome. And that creates a problem of how we get from 1 kilobase to gigabases of DNA. So that's the main thing we're going to be talking about, and to a very large degree, that is a computational problem, and so we need to talk a bit about the computation behind this. Now, originally, when people worked on this, it was done in a more or less non-computational way, and there's a classic experimental technology that was used to try to get from the things you could sequence these short pieces to longer pieces, and that was known as chromosome walking. And the idea behind chromosome walking is that if we can sequence short pieces, then we can use that to kind of step through a piece of genome and get to much larger pieces. So if we can find some starting point, so some piece of the genome we can kind of randomly prime, get an RNA primer to that, then we can use PCR to copy out a piece of DNA and put that through our sequencing machines and get about a kilobase of sequence. Once we've got that, we know what the sequence is for this kilobase of genome, and so we can then find a new primer closer to the end of this. So we can design a primer based on knowing the sequence for the next kilobase here, and we can then do another PCR reaction, get another kilobase of sequence, run that through our sequencing machines, and find a new primer, and basically just keep doing that over and over, walking along the chromosome, hence the name chromosome walking. So this was the original technology people used to try to get from being able to sequence short regions to being able to sequence long regions. And this could work to get you appreciably beyond the kilobases that a sequencer can do, but it did have some significant problems. So one of the big problems with this technology is that it's slow and labor intensive. So there's a lot of work involved in going from running a PCR reaction to putting that through a sequencing machine, getting a sequence, designing a new primer, putting that back into your reaction, get a new PCR reaction, get a new sequence, design a new primer, and so forth. And more than just being labor intensive, it's an inherently sequential process. So basically, each of these steps is going to impose a significant delay, and you can't do all of this at once. You have to do one piece at a time, because you need the result of one piece to tell you how to get started on the next piece. So this is a difficult thing to speed up. You pretty much need to do this one step at a time. You can't what's called parallelize this process and do lots of these things at once. There's also a basic problem with this, which is that you're not really going to get entire chromosomes from this, at least not entire eukaryotic chromosomes. The process will eventually get stuck. And the reason it gets stuck is very important if you want to understand some of the limitations of genome sequencing technologies, and that is the problem of repetitive regions in a genome. So a eukaryote like us tends to have a very large fraction of our genome made up of repeat regions, so small pieces of sequence that are repeated many times in the genome. These might be on the order of just a few bases repeated many times, or called tandem repeats, like we'll see some examples of those later. But these are also things that can be hundreds or thousands of bases, in some cases even larger, but are repeated many times in different positions throughout the genome. And that creates a serious problem for this chromosome walking technology. And we can see what that is if we just imagine what would happen we had a genome that had this repetitive region occurring many times within that genome. So let's say we managed to get a primer and we start our chromosome walking. We get a piece of sequence through our PCR reaction, we sequence it, get a new primer, get a new piece of sequence it, get a new primer, and something is going to go wrong here. So can anyone tell me what's going to go wrong at this point? Yeah, so once our sequence has run into the repetitive region, if we try to prime something that we've read out of that repetitive region, it's going to prime everywhere that repeat region occurs in the genome. So we'll actually get our primer annealing in different places of the genome. Our PCR is going to get a mixture of all of these different strands, and then what comes out of the sequencing machine will be junk. You'll basically get stuck at that point. So this puts more or less a limit on what you can do with this technology. You can get these regions between the repeats, but you're not going to get entire chromosomes. Still, this could give you potentially hundreds of kilobases, up to maybe the order of a megabase at a time. A large chromosome for, let's say, a human being, we might be talking on the order of 50 to 100 megabases. So it's not going to give you an entire chromosome, but you can get an appreciable fraction of the chromosome. So this technology was suggested as a way of getting to a complete genome. And we could take that ability to sequence, let's say, 100 megabases on the order of megabases of genomes, so hundreds of kilobases. Then what we could do, it was proposed, is take our complete chromosomes or our complete genome and try fragmenting this into big pieces. So that means literally breaking up your chromosomes or many copies of them into large pieces of genome on the order of 100 kilobases or 200 kilobases. And then you clone these into some vector, so some organism, typically a bacterium, that will allow you to grow these up and generate many copies of a single piece of genome. So we would get these clones, each of which would have a plasmid in it that would contain one of these 100 kilobase strands. We grow these up, and then you can take one of your bacteria, which has this pure strand, you PCR based on the cloning vector, and you get a sequence of that one strand. You repeat this many times for many strands, and you get on the order of a few thousand or maybe a few tens of thousands of these pieces of genome. Then in theory you're likely to have things covering pretty much the entire genome. So you'll have these big pieces of sequence that you get by chromosome walking. It's a lot of work that goes into getting each one of those, but you do that a few thousand times. And in theory, these pieces will make up your entire genome. There's still more work to do to get the actual genome out of this, and you commonly have to do a version of what's called mapping, which is basically figuring out how to put together these big pieces. There are different technologies for doing this, but more or less the way this works is that you try to find markers, unique pieces of sequence found only at one point in a genome, and then you use these markers to figure out how these large pieces of sequence are put together to produce complete chromosomes. So in the simplest version, you could literally look for the same marker occurring in different ones of these fragments, and then just link together between successive fragments. There are more complex methods where you can estimate approximate distances between markers, even if these aren't exactly overlapping, and then the math gets more involved, but you can put this together. If any of you have taken genetics or are going to take it, you'll see simpler versions of that kind of mapping in genetics, but basically that's the problem that we need to do. We need to find these markers, use these to put together these big pieces of sequence, and then in theory we can go from kilobase things our sequencing machines can do, up to 100 kilobases that our chromosome walking can do, and then up to entire chromosomes that this mapping technology can do. So that was originally proposed as the way we were going to learn the sequences of really large pieces of genome. Are there any questions about this? All right, so this, as I say, was proposed, and in theory this would work, but in practice it really wasn't a good technology for solving this. Even at the time people proposed this as the original way of solving the human genome, it was well understood that this was going to be too slow and too expensive for the technologies available at the time. The idea behind this was that you would kind of start working on this, start doing things this way, and new technologies, new advances would come along. They would bring down the cost and bring down the time needed to do this, and eventually you would get a complete human genome sequence. But in practice, this didn't really happen, and new technologies were needed to actually make it practical to sequence the human genome and to sequence other genomes going forward. So a more efficient technique was needed to bypass the slow steps in this process. And what ended up happening was the development of an alternative technique called shotgun sequencing. And shotgun sequencing is the primary way people actually get large eukaryotic genomes today. The idea behind shotgun sequencing was to try to get around that slow, inherently sequential chromosome walking process by bypassing that, by essentially taking your genome, and instead of getting these big pieces of genome, which gives you a relatively small number you can put together through the mapping process, fragment your genome into much smaller pieces that you can actually sequence directly. So fragment it into 1 kilobase or so regions of DNA, clone those, get lots of little cloning vectors, each of which has a kilobase of DNA, sequence those directly, and then without these intermediate steps, try to put together all these little 1 kilobase pieces. That's going to mean that you will need a lot more of these little pieces, at a minimum on the order of millions of these for something the size of a human genome, but that's the way this was originally proposed. And in more modern versions of this technology, you can actually bypass the whole cloning step. This is no longer needed. You can just fragment your genome, sequence lots of little pieces, copies of the genome, get your little pieces of sequence, and then you have the problem of trying to reassemble your genome from these 1,000 base pieces of DNA. Is that clear to everyone? All right, so this creates a problem, though, and that is that you might now be talking about 10 million or more. Today, this might be getting into hundreds of millions or billions of fragments of DNA that you would want to use in a sequencing reaction. And assembling 10 million small pieces is much harder than assembling on the order of thousands of much bigger pieces, as with the old technologies. And what this meant really is that there was no hope of doing this manually. We had to turn this into a computational problem for this to be something that was doable. I mention this in part because it's interesting to know how the computation works, and it's actually useful if you want to understand some of the problems that occur in genome sequences. So some of the things you can't necessarily trust when you see a genome sequence online. It's also, I think, historically interesting for learning about computational biology, because this was one of the really big successes of computational biology in the history of biology. You really could not have had the genomic era we have today without computational biology, without these kind of assembly algorithms. So this is something that has really radically changed the way a lot of biology is done, and I think that's a useful thing to know about. So I do want to spend an appreciable amount of time talking about how this process actually works. At an intuitive level, it's not so different from what I was showing you with the mapping techniques. We look for overlaps between our fragments, so things that would tell us that one fragment might be immediately before another in the sequence, because if you see a piece of sequence at the end of one fragment that looks like a piece of sequence at the beginning of another fragment, that's at least a hint that maybe the real sequence consists of this first fragment, the duplicate region, and the second fragment in tandem. And doing that intuition, following that over and over, is more or less how you do sequence assembly, looking for these links, these overlaps between these fragments, and then putting together the whole puzzle of joining all your fragments into one big genome. You need more than intuition to make this work, though, so I do want to explain more of the detail that goes into this. So if we're trying to do this, you will look for these overlaps, and it's a bit more complicated. Sequencing machines make mistakes. You will often have multiple repeats between different fragments. And you have to pay attention to the fact that some fragments may be reverse complement from others. Some are from the forward strand, some are from the reverse strand, so you also have to look for that. But the biggest problem is that there is going to be typically a lot of ambiguity. So it may be that the end of this fragment, GATC, happens to be the beginning of this fragment. So maybe the genome consists of this fragment followed by this one. But the end of this one, CGATC, might also be the beginning of this one. So maybe this fragment is followed by this fragment. You don't necessarily know just from looking at one identification of a match between the end of one fragment and the beginning of another that those follow one another in the sequence. We can also have multiple ways you can get to a sequence, so maybe two different ends of fragments match the beginning of this one. There can be a lot of different ways you can put together individual fragments. And in some sense, what we're trying to do then is find a way of linking all of these together that is globally consistent and that makes sense given our data. So find some way of linking end to end between these fragments to get a big piece of DNA using these overlaps between the fragments. Now there may be many ways of doing this that link things together, and we generally need to reason about which are the best or which are the most plausible. And at least intuitively, we would say that a more plausible way of linking these together is one that gets more overlap between your fragments. So more or less what we would pose as the problem we're trying to solve is we're trying to link all of our fragments to maximize the amount of overlap between the consecutive steps of our fragments. So that computationally is more or less what we're trying to do in this sort of problem. So, does that make sense to everyone? And so I want to dwell on this a bit, in part because, as I say, it's important to know how this works, but in part because I think it illustrates a really important set of concepts that I want to get across to you guys over the course of this term, and that is how a computational biologist or a computational person in general thinks about a problem like this. I think this is something that really helps you as biologists to understand what goes into thinking about a problem as a computational biologist and to understand what's doable and maybe why a computational biologist would pursue a particular approach. And this is an example that I think illustrates some of the really interesting principles behind how you think about a problem computationally. In particular, one of the key things that you would often do when thinking about a problem computationally is to try to step back from your problem and abstract it. So we know we've got this big complicated system of assembling fragments to build genomes, but it's actually often useful to try to throw away a lot of the detail and step back and look at a more abstract mathematical representation of what we're trying to do. One of the reasons we often do this is that even if we're working on a problem that no one has solved before in its real-world versions, let's say at some point no one had ever solved this genome assembly problem, we may find that our abstraction of the problem is something people have worked on before. And this was a very important case of that, where we could look at these fragments that we're looking at, and by putting them into the right mathematical abstraction, we can recognize that there is a lot of theory out there on that abstraction. In particular, we used a kind of abstraction very common in computational work, and that is to convert this to a graph. You guys have seen the concept of a graph previously, for example, when talking about hidden Markov models. So a graph is a set of nodes and edges. And in this case, what we could do is convert each of those fragments to a node and create an edge to correspond to these overlaps between fragments. And by attaching weights to these edges that represent how much benefit we get, so how much overlap we get between fragments, we can convert this into a kind of computational problem known as a traveling salesman problem. So a traveling salesman problem is formally the problem of saying that we have a set of cities and we have a salesperson who is trying to visit these cities. And we want to plan a route for the salesperson to visit all of the cities while traveling the shortest distance possible. So maybe the salesperson would want to take this path through this graph of cities. And when we step back to the right level of abstraction, we can see that it's possible to pose the genome assembly problem as a version of the traveling salesman problem. And that ends up being an important thing because these are computationally intractable problems. So I mentioned intractability once before in this class. Does anyone remember what that referred to? So I talked about intractability in the context of sequence alignment, because there it's important to know that pairwise sequence alignment is a tractable problem. We can get fast algorithms that will do that and do it exactly every time, get the best possible answer. multiple sequence alignment is an intractable problem, and so you need a lot more tricks, often a lot more complexity to get good solutions. There's no algorithm that we can find that will guarantee the best possible way of multiply aligning a non-trivial set of sequences every time. And this is an example of another intractable problem, and it becomes useful to be able to recognize this as an intractable problem, and useful to know that other people have already worked quite a bit on that particular problem, or at least a closely related problem. Once we can recognize this, we can see that there is a lot of theory on these traveling salesman problems. A lot of work people have put into them, because many seemingly unrelated real-world problems can be cast as a version of this traveling salesman problem. And we can then go to that theory and bring that to our sequence assembly problem, and by using that theory, get a good first pass that lets us solve this problem on fairly difficult examples, so large numbers of fragments producing large graphs. So that is a lot of why this is important to be able to think about these problems abstractly, and it's also why often in computational biology we need to begin this kind of research by stepping back and looking at a very simplified version of the problem, although you then need to add back in all the detail you threw away because you are dealing with really a very complicated real-world problem. So are there any questions about this? Okay, so I'll return to this a bit later in the lecture, but for now, just be aware that we can come up with these approaches that would give us at least a first pass at solving a genome, so assembling potentially millions of fragments into a genome. This doesn't quite work yet, so there are a lot of complexities of the problem we need to deal with, but even getting around that, even just taking the abstract problem of assembling our pieces of sequence, there are still some significant problems, and in particular problems that have to do with the repeat regions of the genome. So this kind of shotgun assembly technology is actually going to have some of the same sorts of problems, or at least analogous problems with repetitive regions of the genome that we get with the chromosome walking technologies. And we can see why that's the case by taking a hypothetical piece of genome that has some repetitive regions. And I've color-coded this here to say that red would be a repetitive region that maybe occurs multiple times through the genome, maybe many copies of a shorter repeat over and over. And then we would imagine that blue and yellow are unique regions that occur in this piece of the genome. So there's just one blue region, just one yellow region. If we imagine that we've done our DNA fragmentation, then we might get a set of fragments that look something like this. We'll get some purely from the yellow region, some purely from the blue region, some repetitive fragments. We'll also get, hopefully, some from the borders. So some that consist of a little repeat followed by blue, some that consist of blue followed by some repeat, some that consist of repeat followed by yellow, and some yellow followed by repeat. And what we want to do is put these together, hopefully to reconstruct the actual original piece of genome that gave us this set of fragments. The problem is that when you have repetitive regions, it's ambiguous how you do this, and you can actually assemble those fragments into an incorrect piece of genome. So this is another way we can put those fragments together. If we've got long repetitive regions, so much longer than our fragments, and it's the same thing over and over, it's actually possible to misassemble those into something that is shorter than the original repetitive region. Essentially, if you've got the same thing over and over, you would get the same set of fragments from a short version of that as from a long version. So that's known as collapsing repeats. It's one problem that comes up in these kinds of assemblies. We also get problems of misordering, because if you look at the fragments you would get out of this piece of genome, where we have yellow in front of blue, you would actually get the same set of fragments as the one that has blue in front of yellow. You'd still get red and yellow fragments, yellow and red and blue, blue and red. So you can't tell which of these versions of the genome produced that set of fragments. And so this is going to be a problem with these technologies. You're going to have issues around repetitive regions, and you're going to have issues of misordering if you use these kinds of shotgun technologies. They'll work very well for small genomes, so a bacterium typically, which doesn't have a lot of repetitive region. They won't work well for something like a human, which is largely repeats. So does this make sense to everyone? All right, so we needed ways to get around this, and this is a case where you can't quite get around it just through the computation. So computationally, there's no way to distinguish that correct from the incorrect genome assembly, so you needed to kind of change the technology a bit. And this turned out to be a really interesting case where by understanding both the computational problem and the biotechnology, you could come up with a loophole in the biotechnology that made it possible to get around this. And that loophole is something that's become known as paired-end sequencing, originally called double-barreled shotgun sequencing. But basically what this consists of is using the fact that if you can get a piece of genome, what you can do is not exactly sequence 1,000 bases of DNA. So I mentioned before you could sequence fragments of 1,000 bases. What you can do is actually sequence the first 1,000 bases of a piece of DNA, regardless of how long that piece of DNA is. And that's a very important distinction, because if you can get the first 1,000 bases of a piece of DNA, then you can look at the complement of that strand, which is easy to get, and get the first 1,000 bases of the complement of the strand of DNA. So what that really means is that you can get 1,000 bases from either end of a strand of DNA, even if that strand is much longer than 1,000 bases. And that gave you a seemingly subtly different but very important piece of information you could gather, in which you could build libraries of fragments with approximately known lengths. It's pretty easy to sort pieces of DNA approximately by their lengths and, let's say, isolate sets of random pieces of 50 kilobase DNA, random pieces of 10 kilobase DNA, and then sequence both ends of these. So what you then have is not just single 1 kilobase pieces of sequence, you have pairs of 1 kilobase sequences where you know approximately the distance between them, and that distance can be fairly large. And that turned out to be the key thing you needed to make it possible to assemble large genomes that have significant amounts of repeats. So in particular, when we start getting these libraries of paired-end sequences, where we have this big gap where we don't know the sequence, but we know the approximate length, then we have a way of solving the problems that come up from repeats. So if we happen to have, let's say, a paired-end sequence where we have a blue fragment, so a fragment from the blue region joined to a fragment from the yellow region, then by looking at the complementarity of those two ends, we can figure out that blue comes before yellow in the sequence, and we can solve that misordering problem. We can also, by finding paired-end fragments that span across the repetitive regions, get accurate estimates of how long the repetitive regions are and solve that collapsing problem. We can't necessarily get the repetitive region sequenced exactly correctly because we can't really figure out if there are subtle differences between the copies of the repeats which came from one part of the repetitive region versus another, but we can do a pretty good job of figuring out how to order things, what the approximate distances between these are, and solve most of the repeat problem of this technology. So this kind of paired-end sequencing is usually what we're doing to try to assemble large genomes together. Any questions at this point? All right, so this does create new problems for the computation, because even though we've given the computation a piece of information that's now very useful in resolving the problems, we've also broken our nice clean abstraction of how the problem works, and that's going to create all sorts of issues for the theory that we were using to solve the problem. So a lot of kind of tricks go into making this work. You end up with very complicated algorithms. There's a lot of kind of a lot of kind of approximations you need to make to get these things to work, but more or less this gives us a way of putting together computer programs we can use to assemble these libraries of paired-end sequences. It is worth knowing that what you get out of this is not really exactly the right genome sequence. It's not so uncommon that you might have pieces of sequence that are resolved more or less internally to one another, but only approximately know how they're linked together by paired-end sequences. So you may have a good piece of sequence here and a good piece here, but you have an approximate distance between them where you don't know what's going on in between them. You have no sequence. You might also have smaller gaps within these contig sequences, the ones you more or less resolve. And you may have errors in how these are put together. You still will end up with an appreciable number of rearrangement errors, things you just can't place relative to other pieces of the sequence. So usually what we get out of the computations is referred to as a draft sequence, and then there's a process that may take years beyond that of manual finishing, where people will go in very carefully, try to look at these gaps, try to look at possible errors and rearrangements, and kind of fix everything up using all sorts of other technologies to try to resolve the things we can't resolve with the fully automated methods. But more or less, that is how the process of assembly goes. And it's worth noting, when you look at these genome sequences, that these will continue to evolve over time. So the genome sequence we look at today is not going to be exactly the genome sequence in the database a year ago. And when we look at these, when we get into the actual databases, you often need to pay attention to what release of the genome you're looking at, because analysis you do a year ago may have been on an earlier release and may actually not work anymore if you try to apply it to the latest version of the genome. So those are just practical issues you should be aware of in working with genome sequences. Are there any questions at this point? All right, so as I mentioned, there are a lot of real-world computations. What I've talked about is mostly fairly abstract in terms of how these computations work, but we do have a much more complicated system than our abstraction, and you need to pay attention to that. You have to worry about these inexact matches, you have to worry about reverse complement matches. So what you do is really a little more complicated than just looking for overlaps between fragments. You have to do something more like applying a sequence alignment algorithm between ends of your fragments to get these matches. There's also a lot of other data out there, and generally when you're doing a big complicated computation like this, you want to use whatever data you can get your hands on. Every kind of data can help you get a better assembly, but will also make the computation more complicated. So you may have mapping data, you may have other kinds of sequence data. If you're lucky, you may have a homologous assembly. So another version of the genome already solved for that species or a closely related one. And if you have one of those, then you can use it as a template, which makes your assembly much easier, but it also makes a more complicated computational problem. And even at the end, even if you do everything correctly, you've filled in every part of your genome, you'll still have some things that are not entirely resolved. Repetitive regions are going to remain hard to resolve. Even a very well-studied genome like the human genome, we haven't completely assembled what's going on in repetitive regions, and this is probably going to continue for years, and we will keep discovering interesting things in those repetitive regions of the genome. So, for example, genomes tend to be very repetitive around the ends, so the telomeres, and the centers are called the centromeres. People are still discovering interesting things, even new genes, around the centromeres of the human genome and other large genomes. And you may have genuine polymorphisms, so genuine variations between different copies of, let's say, the human genome. We'll be talking in depth about polymorphisms in the next week, but just be aware that this is another thing we need to deal with. Now this, unfortunately, isn't necessarily the end of the story, because as I mentioned in the first week of class, genome technologies or sequencing technologies are constantly improving. It's a very active area of research, and the technologies get better and better every year. So a lot of what's being done today is using what are called the next generation technologies, and those, as I mentioned in the first week of class, can get a lot more pieces of sequence than the older technologies originally used for these genome assembly studies, but they tend to be shorter pieces of sequence. Usually that's a good trade-off. So having 1,000 times as many pieces of sequence, each 10 times shorter, usually is a good thing. You still have a lot more information. But for genome assembly, and especially for the computation behind genome assembly, that's actually a bad thing. It's harder to solve these problems if you have more fragments. So the more nodes you have, the harder it is to solve these graph problems. And it's harder to figure in. You want long reads if you're doing assembly. So there are a lot of trade-offs here, and it's still an active area of research to figure out what to do with these new technologies for assembly. It's still quite common to use not necessarily Sanger sequencing, but versions of the next generation sequencers that give you longer fragments with a lot less of them for assembly, even though that has some significant trade-off. But a lot of work is going on to try to improve the computational models. And I want to mention this in part because it illustrates another important point about how we think about a problem computationally. So even if we have a good approach for our old version of the problem, when the characteristics of the data change, often you need to change the way you think about the problem. You need to change your abstraction, and that's something that's been going on in these assembly problems, where much of the work going on today is actually switching to a very different way of thinking about the problem. And that is something known as de Bruijn graph sequencing, or de Bruijn graph assembly, or Eulerian path assembly. It's basically a different way of taking this problem of assembling a genome and abstracting it into a mathematical model. Still a graph theory model, so a graph model, but instead of taking our fragments, looking at the overlaps, and trying to assemble one of these traveling salesman problems, what we do instead is take our fragments and try to break our fragments up into even smaller pieces. So k-mers, like we saw when we saw the BLAST algorithm, little pieces of K bases of DNA, or K here might be, let's say, 30 nucleotides in a row. And what we do is, we take our fragments, we look at these 30 mers of sequence, and we look at ways of joining these together by looking at pieces where we have a k-mer immediately followed by another k-mer to produce a k+1 mer, so something one base longer. And we can represent that as an edge in a graph. So we take our k-mers, our nodes in the graph, and the k+1 mers tell us which nodes might come after one another in the graph. And we just repeat this going through all of our fragments, and we can end up with a graph that is maybe much bigger than the graphs we were dealing with in our traveling salesman version of the problem. But in this graph, we try to pose a different way of computationally saying what it means to solve the problem. We take this Eulerian path graph, and we try to find a path that now walks through every edge of the graph. If we know that each edge in the graph corresponds to a piece of fragment, then a good solution to our genome should be a path through the graph that visits every edge. So that is known as an Eulerian path. This would be an example of what an Eulerian path looks like. And in principle, if we can find an Eulerian path from this graph construction, we can find a valid way of assembling our genome. Now, it may not be obvious why we would want to pose this in a different way, but when you know enough computer science, you know enough of the relevant graph theory and graph algorithms, you would know that this particular problem has very different properties than the traveling salesman problem. And in particular, even though those seem like they may be very similar problems, trying to walk through your graph and visit every node in a particular way versus trying to walk through your graph and visit every edge, computationally they are very different problems. In particular, whereas the traveling salesman problem is intractable, the Eulerian path problem is actually a very easy computational problem to solve. So it's very easy to take even a large graph and find whether there is an Eulerian path and what it is. Now this does introduce some trade-offs. It helps us quite a bit because it gives us something that will work for very large graphs. So we can take 1 billion fragments of DNA and find Eulerian paths through the graph that they imply. But this does have a significant trade-off with respect to repetitive regions of DNA. So in particular, this is breaking our fragments into these very small pieces. We're typically starting with much shorter fragments than the older technologies anyway. So this will have a much harder time getting around repetitive regions in your genome. This kind of technology will kind of take us back to something we can use for bacterial sequences, but it's not going to work well for something like a human eukaryotic sequence. This is going to get hung up on repeats that are much shorter than the ones that even the traveling salesman versions of this problem could handle. So this is something that's an active area of research right now. This is being used for smaller genomes. It's being used very heavily for assembling small pieces of genomes. So assembling, let's say, the transcript of a single gene from RNA sequencing data. But it's something where it's still kind of an active research problem to scale this up to work for large eukaryotic genomes. But I mention this again because I think it's an important thing for learning how you think about a problem computationally, in particular for understanding how the abstractions you use depend not just on the problem you're trying to solve, but also the data you have available to solve it. Any questions about any of that? Right, so there's a lot still going on in assembly. This is an active area of research, and a lot of things are worth knowing about to give you an idea of what's likely to be happening in the not-too-distant future. As I mentioned, sequencing technologies keep improving. There are now out what are called third-generation sequencers, which tend to give you longer pieces of sequence, but often with some significant trade-offs in terms of error models, the kinds of errors the sequences get that are very hard to deal with. For example, frequent indels, where so an indel would be an insertion or deletion in the DNA, or simply large pieces of DNA that are just missing from the sequence. So there's a lot of work going on to use these. In principle, being able to sequence, let's say, 10 kilobases of DNA at a time will help you a lot with assembly, but it creates hard computational problems to figure out how to deal with these peculiar kinds of errors that occur in these. There are new technologies coming online, such as what's called nanopore sequencing, that may allow us to directly sequence entire chromosomes. That's something that's been, people have been working on this for decades now, trying to get nanopore sequencers working, and it's always kind of right around the corner, but maybe this time it really is right around the corner. But the goal is to be able to eventually get around all of this sequence assembly, although we're not quite there yet. There are diploid sequencers, so people worry now about the problem of trying to assemble, let's say, the two different homologous copies of a chromosome in a single person, and actually figure out what the differences are between those. That introduces other problems. And even more complicated versions of multi-genome sequencing, where we might have DNA samples that have many genomes mixed together. So metagenomic samples, where we might be taking, let's say, a sample of seawater. If you look at your current homework, you'll see there's a metagenomic sequence in that, and you want to assemble pieces of many organisms found from that sample, as well as what's called quasi-species sequencing, where we might be sequencing a very polymorphic, a very diverse organism, such as a virus infecting a particular person. Many viruses evolve very quickly, so if you try to sequence a virus that is infecting a person, you may get lots of vaguely similar but divergent sequences all mixed together, and it becomes a very hard problem to figure out anything useful from that. There's a lot of interest now in single-cell sequencing, so it's become possible to isolate the nucleus of a single cell and use various technological tricks to get a genome sequence of one individual cell. But the techniques that are used to amplify a single copy of a genome introduce a lot of errors into the data. A lot of things are hard to deal with these algorithms, so figuring out how to get useful data out of single-cell sequencing or to assemble single-cell sequences is still a hard problem. But these are things that I think you can expect will be tools that will be available on a fairly large scale by the time many of you are graduating, getting out to working in the field. So just to kind of quickly summarize, there are a lot of technological advancements that have gone into sequence assembly. This has really become a routine part of biological research, a very important part of how it's done. This has become something that has gone from a huge undertaking, even for a bacterial genome, maybe 10 to 20 years ago, to something that's now completely routine for small genomes, and gradually becoming routine even for large genomes like the human genome. Still not quite there yet, but getting there. But you do need to know something about it, because genome assemblies are usually not a perfect inference. There are usually going to be errors, and if you want to work with these, you need to know about the kinds of errors. You need to know that these will undergo a continuing process of finishing. You need to know that they are prone to misordering. So you need to be aware that sometimes things will be rearranged, and that can be quite important if you're working in domains like cancer research, where the rearrangements are often what you're particularly interested in. You need to be aware that some things will maybe not be assembled relative to others. You can end up with what are called orphaned regions, where you simply don't know where it came from. And the sequences will usually have a lot of gaps in them, at least when they're initially assembled, that will gradually be filled in during the finishing process. And it's important to know that repeats are still a problem. We have technologies to get around them, but genome assembly has a lot of problems with repetitive regions, and you usually don't want to place too much trust in a genome where repeats are concerned, and you need to be aware that this is going to be something that's going on for a long time. The technologies are rapidly advancing though, and there are lots of new ideas coming along about how we can use genome sequencing and assembly to solve new problems. I think this is something where as the technologies keep getting better, it's going to be more a matter of creativity than anything else, trying to figure out all the interesting new biology we can solve by applying these technologies in new ways. And hopefully some of you will be the people who think of new ways we can use this to answer new important questions about biology. So we're about out of time here. Are there any last questions about anything new before we break for the day? Okay, so on Wednesday we'll get into some of the issues of what we do computationally once we have this sequence and how we actually get useful information out of it.